Last time by describing the polymer structure of DNA, which had become of great interest to biologists following the work of Hershey and Chase that definitively proved that nucleic acids must be the molecules responsible for holding the genetic code. Based on the work of Hershey and Chase, biologists in the early 1950s had become convinced that DNA was what they needed to look at. But they actually had no idea how it could possibly act as a mechanism for genetic inheritance. Let's begin by stepping back for a second and reminding ourselves of what this molecule has to accomplish to perform this function. It needs to do two things. First, of course, it needs to have some way of providing a code that can store information about proteins. The linear structure of DNA with variable bases along the chain was consistent with the idea that it could provide such a code, because remember, proteins also are linear chains of things, and both of these kinds of molecules are polarized. So you could imagine there could be some sort of mapping of the pattern of one molecule onto the pattern of the other. It wasn't clear what this mapping could be, but they thought they could figure that out, and we're going to return to that code later in the next lecture. Second, the code, the molecule, had to be able to be replicated. If we're going to transmit genetic information, traits, from one generation to the next, it won't work unless we can make duplicate copies of the code so we can hand a copy of the blueprint to the offspring. So the real problem was that it wasn't clear how DNA could be replicated in this fashion. The linear structure of DNA offered some hope for a code, but it didn't offer a clue about how replication might occur. This is where the race, literally a race, for discovering the three-dimensional structure of DNA began. To understand replication, how it might work, scientists were convinced they needed to know the three-dimensional structure of the DNA molecule. And it was this impetus that led to the discovery of the now famous DNA double helix by Watson and Crick. And through that discovery, an understanding of the mechanism of DNA replication, arguably one of the most important findings in biology in the 20th century. And this is going to be our topic today. Now, why biologists should be interested in DNA isn't surprising at this point, given that everybody knew that it was a hereditary material. But why should they be interested in its three-dimensional structure? Well, to understand this, remember back to our discussion of proteins from 2 lectures ago. Protein biochemists had begun to understand in the 1950s that the function of proteins could be understood by figuring out something about their structure. So too it was hoped that some aspect of the function of DNA, and specifically how it was replicated, could be understood by deducing its structure. Now, if this seems like a long shot, it was, but it actually paid off. So what kind of evidence could you use to deduce the three-dimensional structure of DNA? Well, the first kind of evidence came from a procedure known as X-ray crystallography. The positions of atoms in a crystalline structure can be inferred from the pattern that they create when you shoot a beam of X-rays through that substance. Because the X-rays will bend around those atoms, the process is called diffraction, and then when you look at the pattern on the other side, you'll see a pattern of lines and dots, all in particular orientations and spacings, from which, if you're familiar with the procedure and very clever and patient, you're able to deduce something about the relative positions and orientations of the atoms that make up that structure. Now, if you shoot an X-ray through a relatively simple crystal, an inorganic crystal, then you'll get a very regular pattern because the molecules in there all line up in a very regular way. If you shoot an X-ray through a more complex material, like an organic molecule, you get a much, much more complex pattern resulting. And it's pretty difficult sometimes to understand how to pull information from that pattern out and infer something about the way the molecule must be structured. Now, you might say, well, what are we talking about, crystals here or organic molecules? I thought we were talking about DNA. Well, even the most complex organic molecules, in the hands of a good biochemist, can be crystallized. And in fact, that's where this story starts, because Maurice Wilkins, a biochemist working at King's College in London, was able to produce a remarkably pure crystal of DNA. Now, The other person working with Wilkins was a woman named Rosalind Franklin, and she was an expert X-ray crystallographer. She took Wilkins' purified DNA crystal and was able to get what was to that point the most clear and accurate X-ray crystallograph of DNA that had yet been obtained. Now, as I said, these patterns are quite hard to interpret sometimes. Nowadays, we can use computers and algorithms to help deduce the structure of proteins from this kind of data. But back then, it was really pretty much painstaking hand calculation and educated guesswork. As it turns out, Wilkins and Franklin puzzled over their X-ray crystallograph, trying to figure out what they could deduce about the structure of DNA, and they didn't quite figure it out by the time that another person arrived on the scene, Jim Watson, who we're going to get to in just a second. Now, before we introduce Watson, I want to add another kind of clue. There was a scientist working at Columbia University named Erwin Chargaff, who had been for a number of years discovering kind of a peculiar thing about DNA. What Chargaff found was that if you took the DNA from any organism and decomposed it, broke it down into its component nucleotides, you always found a quite interesting relationship. You always found that the amount of the base adenine, which is a kind of a purine, always equals the amount of the base thymine, which is a pyrimidine. Remember, that's a double-ringed and a single-ringed one of these nucleotides we talked about last time. And similarly, the amount of guanine always equals the amount of cytosine. So take apart the DNA of any organism and, essentially measure, count up the amount of the various component nucleotide building blocks, and you always found that the amount of A equals the amount of T, and the amount of G equals the amount of C. This was a very curious relationship that became known as Chargaff's rule. Okay, let's go back to England and James Watson. James Watson was a young American who had just completed his PhD and was interested in protein structure. And he moved to Cambridge, England, and began working with Francis Crick, who was a physicist who was very familiar with X-ray crystallography and how to interpret these kinds of data. They actually were working at the famous Cavendish laboratory at Cambridge University, which is the same place where Lord Rutherford, for example, had first described the structure of atoms long ago. Watson was a postdoc who had come to learn X-ray crystallography and techniques for interpreting them with a general interest in protein structure. Now, the story goes that Watson happened to visit London for a seminar and saw the X-ray diffraction patterns that Rosalind Franklin had obtained from Maurice Wilkins' purified DNA. Watson made some notes, and he rushed back to Cambridge, and he told Crick what he had seen, literally information about the spacing and pattern of the dots on this crystallograph. Using Franklin's data, using her crystallograph, based on Watson's memory and notes, Watson and Crick were able to deduce a number of key structural elements about how DNA must be shaped. Let me list what those were. Now, these are things they figured out by looking at those dots on an X-ray crystallograph, and we don't have to know exactly how they deduced it, but this is what they could have learned, what they learned from that. First, they learned that the molecule had to form some kind of helix. That is kind of a spiral structure, similar to the alpha helix that we saw was characteristic of the secondary structure of many parts of proteins when they fold up. Second, they figured out that the width of this helix, the molecular width of this helix, was about 2 nanometers. That's a really small amount. We don't need to, it's hard to know what a nanometer is. Actually, a nanometer is 1/1000th of a meter, and it's a very small amount. But they deduced what the width of this is. Now, the interesting thing about this is that this width was twice the width of what you would have expected if there was only a single helix in the molecule. And that gave them the idea that there had to be more than one helix, a double helix perhaps. Another thing they learned was what the regular spacing of the repeated patterning along the length of the molecule was. And specifically, they saw that there was a repeated pattern at about 0.34 nanometers, really tiny, which corresponded to the size of about one nucleotide. And then there was a larger repeating pattern that was 10 times that size, 3.4 nanometers. From this, they inferred that the number of nucleotides that would occur when the spiral went around just once and returned to the same point in the spiral had to be about 10 nucleotides. Now, that isn't a lot of information. If I gave you that information, certainly if I had that information and say, okay, tell me the structure of DNA, you're not going to get it from that. And so what Watson and Crick did was to use those data and set out to figure out the structure of DNA the old-fashioned way. They actually literally made physical models of these molecules with stiff metal rods to make large-scale models of DNA. These things stood several feet tall, taller than them themselves. Models that would conform to what they knew the structural characteristics were that had been deduced from the X-ray crystallograph, and also that would have to conform to what they knew about the relative sizes of bonds, sizes of angles, the angles of bonds that would have to make up DNA. I mean, basically the basic chemistry of the DNA. They built many models and asked, okay, when we've got this model, does it all fit together? Does it fit the X-ray data? And does it fit what we know about how these atoms have to fit together? They tried over and over again, and eventually they came up with a model that fit. Now, the trickiest part of the modeling was to figure out how these nitrogenous bases fit into the picture. Remember, a polymer of DNA is a repeating pattern of sugars and phosphates with different nitrogenous bases hanging off the side. Okay, the guanines, the adenines, and so forth. Where did they fit? If you had two or maybe even more molecules of DNA that were spiraling together, where would the nitrogenous bases go? Well, after a couple of failed attempts putting the nitrogenous bases on the outside, Watson and Crick realized they had to go on the inside. Now, why they might want to put them on the outside was the following reason. It's the nitrogenous bases that varied along the length of the molecule. And it's the variation of the different kinds of nucleotides that must somehow involve the code. So if we're going to get access to that code, we've got to make what's different about that code available to the outside world. But they couldn't get the backbones to work together in any way that made sense with the nitrogenous bases on the outside. But if they turned those nitrogenous bases in and had the nitrogenous bases connecting with each other, forming kind of stairs along what's sort of a crooked spiral staircase, with the backbones of these molecules forming what the carpenters would call the stringers that are holding the steps, the molecule began to fit together. And this actually made sense because these nitrogenous bases are chemically repelled by water. They're what we've called hydrophobic. So they want to be on the inside of the molecule. Now, another thing they realized, I'll just mention in passing because this will become important later in our discussion when we talk about replication, is that the two strands of DNA that form this double helix are running in different directions. They're what we call anti-parallel. Remember, I said last time that a DNA polymer is a polarized molecule, and we can define that polarity by talking about the 3' end and the 5' end, which refer to the end that's got an open 3 carbon on that sugar, number 3 carbon, which has a little OH, hydroxyl group added to it, and the 5 carbon end, what we call the 5' end, which has the phosphate group attached to it. So we have these polarized DNA molecules, and what Watson and Crick had figured out was that the two molecules running together in a double helix ran different ways. One ran 3', 5' in one direction, and the other strand, the complementary strand, ran the other way. It would be running 5', 3'. Now, there was one interesting additional problem. And this was actually the most interesting part of this whole story, I think. And that is, how did the bases fit together? I told you that if they turned the bases on the inside, they could get a double helical structure that began to fit the data, but there's still a problem remaining, because remember, there's two kinds of bases. There's the pyrimidines and the purines, and they're different sizes. Purines have these 2 rings, and pyrimidines only have 1. So if you just tried to make these little stair steps across the two sides of the double helix, randomly. You'd have some stairs that are wider if you got, for example, two purines that were big molecules together, and you'd have some stairs that were relatively narrow if you got two pyrimidines, which are smaller. So the outside of this staircase, of this spiral, would be going in and out, which was not structurally stable. And that's where Chargaff's rule came in, because they realized that the implication of Chargaff's rule, the fact that there's always the same amount of adenine and thymine and always the same amount of cytosine and guanine, suggested that it may be that one always pairs up with the other when they're matching together on the inside of the helix. And it turns out when they looked at how these kinds of nitrogenous bases would match up, they found that those particular combinations, A and T and C and G, would always maximize the potential weak bonding, a kind of hydrogen bonding that occurred between those nitrogenous bases. So they'd actually solved two problems. They had figured out how you can have a regular distance along the whole length of the staircase, and also in the treads, and also they figured out what could hold the staircase together, because if you always matched A with T and G with C, you would maximize the potential weak bonding that would hold the two sides together. It's interesting that there's a third property that comes out of that we'll talk about in a second. Now, in April 1953, which is only a year after people became convinced that DNA was the information molecule, Watson and Crick published a very short, a one-page paper in the British journal Nature, which is one of the more prestigious scientific journals in the world, which described the double helix. As I said, arguably one of the most important discoveries in the 20th century. They described the molecular structure, they described how they thought it would all fit together, how it fit the X-ray data, and so forth. But the real significance of this work was not simply to describe the 3D structure of DNA, but instead to show how that 3D structure, to suggest how that 3D structure might actually say something about replication. This paper is remarkable. It ends with the following sentence, which I love. It's sort of British understatement. The last sentence is, and I quote, "It has not escaped our attention that the specific pairing we have postulated," and what they're referring to that is the A with the T and the G with the C, "immediately suggests a possible copying mechanism for the genetic material." The fact is that the fact that A and T and G and C were always paired together meant that if you took the two sides of the molecule apart, you would always know what the other side have to be. We're going to talk about that in a second. That's called complementary base pairing. And it was this fact that suggested the mechanism by which DNA was replicated. Now, just later that year, in a subsequent longer paper, Watson and Crick elaborated on this copying mechanism. And what they showed was that, what they suggested was that it was this so-called complementary base pairing where A and T, and C and G were always matched together, that meant if you could somehow unzip the molecule, take it apart, and have the two halves separate, you could always retrieve the sequence in the complementary half that had been removed. You would always be able to say, if there's an A on this side, then the other side must be a T. There's a C here, the other side's got to be a G, and so forth. Now, they proposed then that what happened during replication is that the old DNA strand, when these two, an old DNA strand could serve as a template for two new DNA strands by having the old DNA strand separate and having the opposite sides built on to the existing strand, creating two new DNA strands. And thus, the information in DNA would always be duplicated exactly. And at the end, we'd be left with two double helices that were copies of the original single double helix. Now, I want to stop for a second, or pause for a second, and give you a little bit of a historical note. As you can imagine, this was a very important discovery, and it was awarded a Nobel Prize, the Nobel Prize for Physiology and Medicine in 1962, was awarded for the discovery of the double helix of DNA. Who got the prize? Watson and Crick, of course, because they had published the paper that described the structure, and also Maurice Wilkins, who was the biochemist who had purified DNA. Interestingly, Rosalind Franklin, the woman who actually made the crystallograph, which Watson had seen and which provided the essential information for understanding the structure, was not awarded the prize. This has been the source of some interest and controversy over the years. Why not? Much has been written suggesting that, in fact, Franklin was denied credit for what was probably this most important contribution of the double helix. Certainly, it was well known that her relationship with her colleague Maurice Wilkins was a little bit rocky. Wilkins assumed that when Franklin showed up to work in the lab that she would be more or less his assistant, and that wasn't her attitude. She had a very difficult social interaction with most of her co-workers, and she was eventually asked to leave the lab. And she was told at the time by the head of the lab that she should stop her work on DNA. Franklin did actually publish one more paper on DNA. It turns out it was a paper that helped confirm the Watson and Crick theory. But then she put down her work on DNA and she began a completely different career, using her X-ray crystallography skills to look at things like the structure of viruses and so forth. It's also true that if you look at accounts of the discovery of DNA, say in the 60s and the 70s, the contributions of Rosalind Franklin are relatively understated. But the fact that Franklin did not share the Nobel Prize actually has to do with something other altogether. And the fact was that she had died before it was awarded. Nobel Prizes aren't given posthumously. Franklin died of ovarian cancer in 1958 when she was only 37 years old, 5 years after the description of the double helix by Watson and Crick, and 4 years before the Nobel Prize was awarded to Watson, Crick, and Wilkins. Fortunately, in recent years, a number of biographies and other kinds of historical analyses have made it clear what the critical importance of Franklin's contribution to this work was, and she now figures prominently in just about any textbook or description of the process. Okay, well, let me put that aside, and I want to go on, and I want to go back now to this proposal that Watson and Crick had for how the double helix might be replicated because of this complementary base pairing. Watson and Crick suggested that the molecule must unzip, and then each half of the molecule could serve as a template for a newly formed half. It's a good hypothesis, but was it correct? Actually, as soon as Watson and Crick proposed that, there were two alternative hypotheses very quickly proposed. The first alternative suggested no, the DNA double helix must remain completely intact when it's replicated. That is, the two strands don't separate, and the entire molecule is used in some fashion as a template for making more DNA. This alternative was called the conservative hypothesis of replication, or the conservative model of replication, because the original DNA double helix molecule remained completely intact and conserved. The idea must be that there was some intermediate molecule that got information from the structure of the DNA helix and used it to build a completely different helix. Now, a second alternative suggested that the original DNA molecule becomes completely broken down during replication, with the newly copied DNA in the right order assembled by some unknown mechanism. In other words, the DNA double helix in this model would actually be irrelevant. It didn't tell us anything about how we might copy DNA. It just happened to be a double helix. This alternative was called the dispersive model. It was called dispersive, obviously, because the DNA in the original helix that's copied just becomes dispersed and incorporated wherever in the new copies that are created. Now, based on what was known about molecular biology of DNA in the 1950s, both of these hypotheses were reasonable as any other, although neither had as neat a solution to the problem of duplicating the exact order of the nucleotides in the strand of DNA, this order being the information that we're seeking. Now, in hindsight, knowing what we know about DNA today, both of these alternatives actually seem quite unlikely. But their value then, and still now, was to serve as alternative hypotheses against which to test specific predictions made by the Watson and Crick model. Because what we now want to do is to go beyond the hypothesis that Watson and Crick had about how DNA was replicated to testing specifically whether that hypothesis was true. The mechanism, by the way, that Watson and Crick proposed became known as the semi-conservative model of DNA replication. This was called semi-conservative because it predicts that during replication, the double helix unwinds, and now the new daughter helices will both have one strand of the old helix. It's important to keep in mind. We begin with one helix, it separates somehow, and the resulting daughter helices that are formed maintain the original halves of that parent helix, upon which, or opposite which, new halves are formed to create the new double helices. This hypothesis led to a specific prediction. The prediction was that if you could know which was old and new DNA after replication would occur, all the old DNA that was in the original parental double helix would now be dispersed between the two daughter helices equally. And the daughter helices would be composed of one half of old DNA and one half of new DNA. Now let's contrast that to the predictions we might have if we looked at these other two models. Think about the conservative model first. That model suggests that the DNA helix just remains intact. Once you've got it, you've got it. After replication, that model would suggest that the two daughter helices would be separately made up of, on the one hand, all old DNA, and on the other hand, all new DNA. In other words, the old DNA in the original parent is still in the original parent because the original parent is just copied intact, and the other daughter DNA helix is now completely new. The dispersive model made yet another prediction. That prediction was that the old DNA that was found in the original parental helix would just be randomly scattered, as its name suggests, across the two daughter helices. So we have three specific and different sets of predictions that could be used to distinguish between these three models of replication. The trick is figuring out how to know what's old and new DNA. Actually, it was several years, it was a number of years after Watson and Crick's original proposal before anybody could figure out how to experimentally test it. I mean, bear in mind that at that time, this was the hottest question on the planet, really. How does DNA replicate? Watson and Crick, through this remarkable model of the double helix that they had developed, had come up with a very neat hypothesis, a hypothesis that, if it were true, would provide a remarkably elegant molecular way to achieve this most important thing that a biological information molecule has to be able to do, and that is to accurately make more copies of itself. And the way to test this hypothesis was pretty obvious conceptually. Let's ask ourselves, after replication, what has happened to the material in the original parent's helix? We have very distinct predictions. The problem was figuring out how to know where old and new DNA was. This is often the way it is in science. An idea comes forward, people understand what they have to do, but the critical experiment isn't actually done until somebody comes along and says not, what's the experiment, but how can I actually do this experiment? And sometimes that can take years, as it did in this case. Eventually, researchers figured out an extremely clever way to know what the difference between old and new DNA is. And that's where we're going to begin next time with testing Watson and Crick's model.